and pass over to Louisa. And I think we're going to start with a little video encapsulating her day, and then we'll go on to some questions. So I'll be back very soon.
Yeah, great. Hi, everyone. My name's Louisa, and I think hopefully we're going to play a video I'm going to giving you a tour around my building and showing you some of the labs that I work in. So yeah, I'll happy to take your kind of questions after the video, but we can watch that first.
Hi, everybody. My name's Louisa, and I'm a scientist from the Biodiscovery Institute at the University of Nottingham in the UK. My research focuses on brain tumour research, specifically looking at drug resistance in brain tumours. So today I'm going to give you a tour around the Biodiscovery Institute and show you a bit more about what we do. So this is the reception of the building that I work in, and one thing I wanted to show you before we go and look around some of the labs is our food bank. So here in the Biodiscovery Institute we're really focused on kind of links with the community and supporting our local community. So one thing we have here is a food bank drop-off point, so we all drop off small items of food as often as we can, whenever we can, and then it gets dropped off to our local food bank in Nottingham every couple of weeks to a month. So it's a really lovely initiative, and yeah, one that we're really proud of. So welcome to one of our cell culture suites in the building. So as the name suggests, this is where we culture or grow our cells. So as a cancer research scientist, I have to grow cancer cells in the lab. And this allows me to do things like test how the cells respond to different anti-cancer therapies. And this is really important when we're looking at improving the treatments that we give to patients. We need to test everything on cells first. So we grow the cells in the labs in little cell culture flasks that you can see here. So the cells are all here, and my cells are attached to the bottom of this flask, and then the liquid you see is media. So it gives the cells everything they need to grow, all the nutrients that they need to keep on growing. So if you come around here to the screen, hopefully if I focus it for you, you'll be able to see some of the cells here. So these cells I always think are quite cool because they grow like kind of countries. They look kind of like continents if you look on the screen, which is quite good. So you might be wondering where we keep these cells when we're not looking at them. So if you come over here, so we keep our cells in things called incubators. So if I open it up, you'll be able to see all of the different cells in the different sizes and shapes of flasks inside. Now, one important thing to note about incubators is the temperature. So incubators are always set to a temperature of around 37 degrees, which you hopefully can see up here. And this is because the human body is actually quite hot inside. So the human body is 37 degrees. So to keep our cells growing like they would in the human body, we need to make sure that our incubators are set to the same temperature. So how do we do any experiments in our cells? How do we grow them? We use safety cabinets, so you can see one here. So this is a cell culture hood. The gap here is where you put in your arms where you're doing cell culture. And this is important because this means that I cannot breathe onto the cells, so I can't infect the cells with any bacteria that I'm carrying on myself. But it also means that anything in the cells can't get to me, so it keeps both the cells and me safe when we're working on them. So the next thing I wanted to show you is where we store our cells when we're not using them. So we want to be able to use our cells in the lab, but then sometimes we'll be having a break between experiments. So we want to be able to store cells without them getting any older. So one thing that we can do to do this is store them in something called liquid nitrogen, which you might have heard of. So liquid nitrogen is a cryogenic liquid, which means it's really cold. In fact, it's 196 degrees, so minus 196 degrees. So it is freezing. So we do this to kind of freeze our cells, it stops them growing, it stops them developing, and then we can recover them at a later date back into the lab to get them to start growing again. So basically, we kind of treat our cells like they did in Han Solo in Star Wars, if you've ever seen that. So they freeze him and then he comes back to life and hasn't aged. That's what we do to kind of thaw our cells and be able to keep on using them for years and years and years. So this is our main molecular biology lab in the building. So as you can see, it is full with lots of different pieces of equipment. And this lets us do all the different experiments that we need to do every day to answer the research questions that we're interested in. So I'm going to show you one piece of equipment that I use pretty much every week, and this is called a real-time PCR machine. And this little machine is what we can use to look at gene expression. So if you don't know what your genes are what makes you, you. So they control your eye colour and your hair colour. So they're really important. And if you're looking at diseases like I do, it's really important to understand how gene expression changes in those diseases, as this can allow us to design more personalised treatment for those patients. We can understand how their gene expression levels change. So I thought this would be quite a cool thing to show you, just to show you the tiny scale that we do these kind of experiments. So this is the plate I use that fits into this machine, and it has 384 tiny wells. So as you can imagine, you need to have quite a steady hand when you're putting together these experiments. Each well is its own individual reaction. So this means we can get a huge amount of data from one kind of plate that's just the size of my hand. So once I plate these plates, I just open the machine and I just place it in, making sure it's the right way up. Then we just close the machine again, and then we can set it off, and the machine takes about 2 hours to run, and after that we can get a huge amount of gene expression data. So this is a really important piece of kit. So to do all of these different types of experiments, we obviously need lots of reagents. So we need lots of different mixtures of different things to help us find out all these research questions. So we have to store these things at different temperatures. So we have some basically freezers, but they're set to -80 degrees, so they are very cold. And this is where we keep our more sensitive samples and reagents that would degrade at higher temperatures. So if you follow me, so here we have our cold room and our freezer room. So the cold room is basically a massive walk-in fridge. So if you come through here, this is where we keep all our reagents that need to be stored at fridge temperature, which is 4 degrees. So it's quite cold in here, you don't want to spend too long in here. But what's even colder is the freezer room. So this room is where we keep everything that needs to be stored at -20 degrees. And as you can probably tell, it is really cold, so you definitely don't want to be staying in there too long. You want to have well-organised boxes, so you're not staying looking for things for ages in there, because it is very cold. Right, so we're going to get out here now because I'm freezing. Thank you so much for joining me on my tour around the building today. Before we finish, I just wanted to talk to you a bit about my kind of journey into science and how I started working here and became a scientist. So I actually never wanted to be a scientist. When I was younger, I wanted to be a vet. So from about the age of 3, I had my heart set on becoming a vet. So I did work experience on farms and in stables, in kennels, and worked really hard to get an application together to go to vet school. But unfortunately I didn't get in, and I also didn't get good enough grades to then reapply the following year. So I managed to get a place on a course in Nottingham studying biochemistry, and at the time I wasn't that excited to go. It wasn't really what I wanted to do. But as soon as I did my first couple of lectures, I just loved it. And the more I carried on into the degree and got to do lab work and research projects, I just realised it was definitely what I wanted to do. I loved being in a lab, I loved being in control of my own research, and that really pushed me to then apply to do a master's project and then a PhD, which I'm doing now. So a PhD is a 4-year research degree, and at the end of it you get to call yourself a doctor, which is pretty cool. So again, things didn't quite go to plan. So I applied for 12 PhDs and actually got rejected from all of them. So again, I had a few kind of road bumps, but I kept on applying and I kept on going, and I got there in the end, and now I'm in the final year of my 4-year PhD. So one thing I just wanted to say is, even though things don't always go the way you might have hoped or you might have planned, just keep going, keep trying, and I promise things do always work out in the end. So thank you very much for joining me, and I'm looking forward to taking your questions later. Thank you.
Thank you for that, Louisa. That was such a brilliant video, and I really loved hearing a bit more about your background as well. And I think that's such an important lesson, that life doesn't always go the way you've planned it, and there is detours and speed bumps and obstacles. But honestly, that is part of what makes it so fun, and you fall into things that you never would have expected, and you end up absolutely loving them. And I think that's such an important message to get across. Now, we're lucky to have Louisa with us for a little bit longer today to answer any of your questions. So again, the easiest way to ask is to go into the Q&A section. And if you look for that little triangle, square, circle button, you can click on that and then go into the Q&A and type your questions there. If you've got any questions about her research or her job or her journey through academia to get there, then please go ahead and pop those in the chat just now. Just while you're having a think and giving you a second to type your questions in, I maybe start the ball rolling. So you were saying you were doing your PhD at Nottingham. Is that where you did your undergrad as well?
Yes, so I did a 4-year undergrad, and then I stayed on and did another master's after that, which is another research degree. And then I stayed on, obviously, for another 4 years to do my PhD. So I've been at Nottingham for a very long time now. But I've changed projects quite a lot. So when I started, I was actually studying cancer, but I was using fruit flies as a model organism. So I did all my experiments on tiny, tiny flies that are like this big. And we use them because although they are obviously look very different to us, a lot of the genes in flies at a disease level, so the ones that we're interested in if we're looking at cancer, are the same in humans and flies. So we can use them as what we call a model organism. So yes, I did lots and lots of experiments in flies, and yeah, lots of things like that, which is very different to what I do now, but it was a really good experience. I did lots of like tiny dissections where you have to be really, really exact. But yes, it made me realize that I really enjoy cancer research when I did my first kind of projects in the fly labs looking at cancer. So now instead of using flies as a model organism, I use cancer cells instead. But yes, so I still do cancer studies, but kind of a different model and a different cancer I'm interested in.
Nice to have that opportunity to try out different things as well. We've got some questions in the chat here. I'm just going to work my way through them. So the first one is saying, how long did you study for your first PhD, or how long does a PhD take?
So a PhD can last anywhere from about 3 years upwards. So I'm doing a 4-year one. So it's a bit longer, but I've got to do an internship as well. So I got to spend a few months doing something, and they asked it to be completely unrelated to what I do in the lab. So I couldn't just go and work in a different lab. I had to think a bit outside the box and spend a few months doing something completely different. And it was a way for us to kind of work on our social skills and find other things that we're interested in outside kind of academia and science. So I actually spent that organising a science festival, a bit like this one, but a lot smaller, in Nottingham. So I did that for my internship. So my PhD is a bit longer because I did that, but yes, 4 years.
Oh, I'm glad you had that experience of doing science communication as well, because we've definitely benefited at TechFest from being able to grab hold of you from that side of things and have the opportunity to have you come and speak with us today. So very pleased that that was something that you were enjoying as well. I've got 2 questions here that I think are kind of complementary, so I'm going to ask them together. So Clooney P6 and 7 were asking, is your job dangerous? And then we have another one that's sort of linked to that, that's saying, is it scary working in a lab?
So my job personally isn't very dangerous at all. So although I have to wear, so you saw in the video when I was in one of the labs, I had safety goggles, and I also always have to wear a lab coat and gloves. And that's because I use certain chemicals that might hurt if I ever spilt them on my skin or got them in my eyes. So although we have to wear kind of protective equipment, there's nothing too dangerous doing what I do. And it's not particularly scary either. No, the couple of things that we do that might be a bit more scary, I guess, is so when I showed you the massive tank where we store our cells, which is full of liquid nitrogen, I do a few experiments where I need to pour liquid nitrogen and have that separate. So it's not in a tank, it's kind of in a smaller kind of container. And I use it to freeze things really quickly. So if I want to freeze something super fast, I drop it into a big container of liquid nitrogen. And because that's really cold, you really don't want to spill that on yourself. So that's maybe a tiny bit more scary, but yeah, no, nothing's too scary that I do in the lab, thank goodness.
I guess so long as you have the right health and safety and you're following the rules, then it's okay. But if you guys haven't seen liquid nitrogen before, it's super duper cold. So yeah, you wouldn't want to make a mistake with that. Oh, we've got quite a technical question here. That says, is it possible to reach absolute zero? I guess this is because we were talking about the different temperatures in different freezers and fridges.
So liquid nitrogen is minus 196 degrees. Absolute zero is colder, I think. Is it like minus 200 and something, minus 270 something off the top of my head? And people are interested in absolute zero because it's when, I think, if you're looking at thermodynamics, so I think it's when you have the lowest amount of energy in a system, but it's not actually an area that I work on at all. So I'm not sure about how you reach it and how you work at that kind of temperature, but I'm afraid. But yeah, sorry.
That's
I'm not sure if I can
really, really cold.
very cold temperature.
That's a really good question, though. Maybe that's something that we could look into some more. Well, we got a question from a P2 class here that's asking, is it cool being a scientist?
I think it's cool being a scientist.
I
No,
think it's cool
I
too.
love I love doing it. I think I've always kind of really liked kind of problem solving and things like that. And I think that's really all science is. It's just you think of a question that you think kind of, or a problem that you really think needs a solution. So for me, when I'm looking at, I study brain cancer. So we know that some of the treatment needs improving. So that's kind of my problem. And then you just work on kind of solving that problem and answering that question. So I always think if you quite like kind of board games and things like that, like Cluedo, that kind of, yeah, question answering and problem solving, then you'll really love science. And yeah, I think it's really cool. You get so many different opportunities. I think something that people don't really realize is how much you're allowed to travel as a scientist. So we do conferences all around the world, which obviously is paused at the moment because of the pandemic. But yeah, we do international conferences. So I was supposed to go to Japan last year for a conference, which would have been great. And I'm hopefully going to Boston at the end of this year. So we have loads of really cool, amazing trips that we can build into our research. And you get loads of opportunities to do things like this as well, which is great. So yes, I think being a scientist is a very cool job.
Oh, I love that. I think it's super cool as well. And I really like that idea there of if you're not really quite sure what a scientist is or what it does, I like that idea of if you're somebody that enjoys puzzles or problem solving or is just really curious about the world, then maybe science is something that it's worthwhile for you to look into a little bit more. The next question I've got here says, how long have you worked as a scientist?
So I started, I guess, during my undergraduate degree, which I started in 2012, I think. So it'll be about, I guess, 9 years now.
That probably seems like a really long time to a lot of the people watching. And to me, I'm like, oh, 9 years. That's fine. It's still early in your career, but so next question in here is a really interesting one. It's a really good question. It says, in the first lockdown, did you see a difference in the plastic waste? And I know that that might not necessarily be your area of expertise, but it's maybe still something you could comment on, or even just looking at anything that you've sort of been able to see from as a result of the first lockdown.
Yeah. So if in terms of plastic waste, that's a really good question. I think, well, it's quite interesting actually. I think if you think about science in general, I don't think you realize, but we use a lot of plastic. Everything we use is single use. So that's a very good comment, is how can we improve as scientists and make our work a bit greener? Because as you saw in one of the first films I showed you where we grow our cells, once we've used something on our cells, if we use like a pipette, which is how we transfer bits of liquid between the flasks, then we have to throw it away straight away, and everything's plastic and we can't recycle it because it's touched kind of human cells. So it's a really, really big problem in science, is the amount of plastic waste that we produce that we can't properly recycle. And it's definitely something that I think people will start looking into, is how to make science and kind of medicine in general a lot more green and a lot more kind of sustainable. I think one difference I did see, certainly in the lab looking at plastic waste, is because of the lockdowns and combined with Brexit, obviously we haven't been able to get a lot of equipment. So I think our plastic usage has definitely actually gone down massively, but that is mainly because we haven't been able to get like nearly as much equipment and reagents and the things we use for all of our experiments. We haven't been able to really get as many as kind of packs of things as we used to. But yeah, no, really good question. Definitely something I think that needs to be explored more in science.
Yeah, that was such a good question. And I hadn't really considered that before, that the plastic that you're using in the lab, you might not be able to recycle because of it might be hazardous or have touched hazardous materials. So that seems like a really good problem that hopefully somebody's going to start working on at some point. And if hopefully by the time you guys are all grown up and going to university, then that'll have been solved already. But if not, then maybe we have the answer watching us just now at the moment, and one of you guys were going to crack it. Um, Next question I've got here says, what made you interested in working specifically in cancer research?
So I think when I started my first degree, I wasn't quite sure what I wanted to kind of study at all, really. But as soon as I went to kind of the cancer lectures, I found them really fascinating. And my father had cancer. I think a lot of people know someone who's had cancer. So I think you definitely kind of feel that there is a real need to research kind of that area and improve treatment. So I think when I learned about it more and more, not only did I find it really interesting and the process is interesting, but also I think it was the thought that I could potentially help people like my dad, and I could do something that would really, benefit other people. And that's what made me really kind of continue with working in cancer research, is that hopefully one day I might make a discovery, however small, that might help. Even if it's just like one or two people or a few patients, that would be really amazing. So that's kind of the drive that makes me continue in cancer research.
Oh, that's such a lovely answer. And it just fills you with so much hope when you think about all the people all over the world that are all sort of coming together and breaking down these barriers to work together to try and help us solve some of these problems. It just seems like it's such an important community to be a part of, and everybody that contributes, whether you're making giant leaps or just tiny little steps forward, it all adds to the whole. And so we're so lucky to have scientists working away to come up with solutions, whether it's medicines or dealing with climate change or some of the other things we've touched on. It's such an important role that they play in an important profession. Oh, next question I've got here. Okay, so how long would it take you to get hypothermia in the freezer room? I think this is the -20. How long can you be in there before you're starting to be in danger?
I don't think probably very long, because in the lab, our labs are actually very hot. So we just wear like t-shirts under our lab coats. So we're never actually prepared to go into a room that's -20. So, I mean, I get freezing. If I have to spend more than a few minutes in there, I really, really struggle. So obviously one thing, so you have to close the door behind you to keep the temperature in. One thing we have to be really careful of is we have big alarm panels down next to the exit. So if you ever do get locked in, or the light switch unfortunately is on the outside in the corridor. So if someone ever accidentally turns off the light and you're stuck inside, we have lots of like big alarm buttons that you have to press, and then it sends an alarm around the entire floor, so then someone can come and make sure that you get out. So we do have things in place to make sure that never will happen.
I guess it's good that the alarm's there, but you wouldn't want to risk it anyway. -20, that is so cold. Although I guess it's a good excuse to make sure you keep everything organized.
Yeah.
Maybe I should start pretending that for my freezer at home, so I can start being a little bit more organized and actually find what I'm looking for. The next question is kind of a follow-up for that of what is the coldest temperature in the lab? So we've talked about the -20 freezers. Is that the coldest freezers that you've got?
So we have -80, which is a kind of a stand-up freezer as well. So you can't walk into those, obviously, because that would be very dangerous. But we do have -80, and that's where we keep things like, we can keep cells in there, and really, really sensitive samples that if they get a bit warmer, they would start to kind of break down. So if we're looking at things like RNA and so DNA, the things that kind of make up our bodies and control our kind of what we look like, that kind of make us us, that's actually very temperature sensitive. So you want to keep it at really, really cold temperatures to stop it breaking down. And so -80 temperatures let us store things for a lot longer than we would be able to store them if we were using kind of a normal freezer. So your normal like home freezer is about -20, I think -17. So yeah, we also, obviously the liquid nitrogen is probably the coldest. So that's -196. So that's definitely the coldest. So yes.
It's very cold. Oh, this is a fun one. Okay, so I've got another question here that's asking, what's your favorite experiment? And have you done any experiments that have gone wrong?
So trying to think what my favorite experiment is. I'll answer the second part first. So have anything's ever gone wrong? Yes, is the answer to that. I think as a scientist, when I started kind of doing research, I honestly thought that it was, you just kind of designed an experiment, you did it, you found something out. And then as I've kind of progressed and got more advanced in my career, I've realized that everything goes wrong all the time, and the majority of science is really kind of troubleshooting and adapting things and making them work a little bit better every time until they do finally work. So I think every experiment pretty much that I do goes wrong the first time, and then I have to kind of redesign it and make it work better so it works the second time or the third time or the fourth time. So yes, lots of things go wrong, but I think it's part of being a good scientist is being quite resilient and being able to change your plans and adapt to situations. I think that's really, really important if you want to be a scientist. And also you have to be very good with kind of failure and things not working. And then my favorite experiment, I love doing anything I can working with cells. So we do lots of kind of experiments in our lab where we're interested in how cells can invade and how they can move. So we do things where we make kind of a gel, and then we put cells on top, and then we see how they can kind of migrate and invade through that. So that's how we can look at invasion in things like cancer and other diseases where cells move from one part of the body to another. So I think anything like that I find really, really interesting. Anything where I can use the human cells and kind of make a really cool model for them to work with, I think is really fun.
That's such a good answer, and I really like the idea of actually there's quite a lot of failure or quite a lot of things that have gone wrong, and I think that's kind of applicable across the board, and for a lot of you guys as well. It's a good lesson there that things won't always go right the first time, and that's okay. And what's important is to just keep trying and figure out, well, why didn't it go right, and what could we do different next time? And I think no matter what you end up doing, that's quite an important lesson. So thank you for that. Okay, I've got another question here. So we've talked a lot about liquid nitrogen. It's very much capturing you guys' imagination, which is what we like to see. But there's a few people that are worried about your safety. So they're asking, how dangerous is it? Like, if it touched your bare skin, would you be in trouble? And what can you do to keep yourself safe?
So if it touched your bare skin briefly, you would just have kind of like an ice burn. So I don't think it'd be painful, but it wouldn't be very dangerous. I don't know if you ever, it's something that you could do in your classes. You can, if you like Google on YouTube, if you looked at liquid nitrogen experiments, you can do lots of really cool things where you can dunk like even like say a bouquet of flowers, like a bunch of flowers, you can put that in liquid nitrogen. If you take it out and hit it on the ground, it will shatter. So that is how cold it is. So obviously if you ever to put your hand in it for too long, then that would be very, very bad. So you never want to actually, submerge yourself in it. But so it's very dangerous if not used safely, but we use it very safely. We have thin gloves, then thick gloves over the top. We have goggles and a visor and a lab coat, so we are perfectly safe when we use it.
Glad to hear it. I think there's just a lot of people picturing how cold that might be, and you definitely wouldn't want to touch it. So no, it's good to know that all the proper procedures are in place. I know we're coming up close to your guys' home time. So we're going to start losing people, and I've gotten through the questions that are there. Do feel free to add any more in the meantime. But I thought just before you have to disappear off, I would ask a question of my own to Louisa, which is, would you have any advice to any young sort of budding scientists or younger kids that are maybe not sure what they want to do next? But what advice would you sort of have for them?
Yes, so I think the first thing I would say is that there is definitely an area of science for everyone. So although maybe I do kind of the more biology side of things, if you find maths interesting and you like maths, you're good at that, then there's kind of things like physics. If you're interested in how things work and designing things, then you can do things like engineering. So there's really something for kind of every interest. And that's why I think it's such a great thing if you can just go into like, maybe when you're picking kind of your exams and what subjects you want to take, if you just have a few sciences in there, it will open so many doors for you. So I would say my first kind of advice would just be to give it a go, because I think there really is something that everyone will find interesting in science. It is really, really diverse and really fun. Another thing I would say for someone wanting to get into science is I think as I said in my talk, just don't be afraid if things go a bit wrong to start off with. A lot of science courses can be quite competitive. And as you heard with me trying to apply for university and then PhDs, I got rejected from so many. So I've never actually been properly accepted onto anything. I only got my PhD. I was actually on a wait list and someone dropped out, so I got it. So I've never got accepted onto a course, but you just have to be really persistent. Make sure you get yourself out there, introduce yourself to people. If you're applying for a course at university, email professors, find out about the courses. So I'd say be really proactive and never give up, because if you just keep going, you will get there in the end. Like I did, it took a while for me to find the right PhD and the right course that I wanted to do, but now I couldn't be happier, and I feel so lucky to be able to do what I do every day. So yeah, my main thing would just be to never give up and just keep trying.
Such brilliant advice, and I think that's a really nice note to end on. I know we're almost at the end of the day on a Friday, so a huge thank you to all of you guys for joining us. I want to say a massive thank you to Louisa. That was so brilliant and such a lovely insight into what you do, and it's lovely to hear you speak so passionately about it as well.
Oh, thank you for having me.
No problem at all. If you have any other questions that we've not covered, that you realise, that you think of later and you think, oh gosh, I wish I'd had the opportunity to ask that, just email us again. Ask your teachers to email TechFest. We'll pass it on to Lisa, and I'm sure she'll be happy to get back to you if there's any other questions that you think of. This is sadly the last day of our Digital Festival live event, but don't panic. If there's anything that you wish you'd seen and you didn't get the chance to, or if you go back and look at the programme now and think, oh no, I wish I'd been able to see that, a lot of our sessions have been recorded, a lot of our brilliant shows and presentations have been recorded, so you will find them on our YouTube channel, which is TechFest Aberdeen. You'll find a lot of our content there. So once again, thank you very much, Lisa, for joining us today. Thank you guys for coming along. We hope you enjoyed today's session, and hopefully we will see you all again very soon. That's bye for now.